Hey everyone, thanks for coming to my talk, Early Life Interactions with Aquatic Insects Elicits Physiological and Behavioral Stress Responses in Lake Sturgeon. So I investigate stress using lake sturgeon as a model system. This is a fish species that's threatened in the Great Lakes, so they're a priority for conservation. Their populations are vulnerable to environmental stressors, and they also have 99% mortality during the first year of life, especially these early life stages. A lot of this is due to predation, and so my experiments are focusing on these early life stages. This study was conducted at the Black Lake Sturgeon Rearing Facility in Onaway, Michigan. So we know that interspecies interactions are really important influences during these early life stages, especially if that involves interacting with predators. During the free embryo stage in lake sturgeon, they are burrowed into the gravel substrate, living off of their yolk sac reserves until they reach the larval stage. They share that habitat with a diverse community of aquatic insect larvae, some of which are predators. So isonychiids in the top picture are filter feeders, and then perlids in the bottom picture are predators. For this experiment, I used free embryo sturgeon, all from the same family, divided among 3 treatments for 3 days to see how they responded to interactions with these aquatic insects. The control treatment had no insect. The isonychiid treatment had 4 insects added to each of the replicate tanks and left for 3 days. And then the perlid treatment had 4 perlids added to the lake sturgeon tanks to interact for 3 days. At the end of the 3 days, the sturgeon had reached the larval stage, and that's when I collected all of this data on mortality, cortisol, and behavior. For the behavior trials, I took 6 individuals from each replicate tank and put them in a petri dish and recorded their swimming activity for 4 minutes. After 4 minutes, I added an odor to the center of the petri dish and continued recording those same individuals for another 4 minutes. The odor that I added was made from dead sturgeon larvae, so it contained the alarm cues that they react to as a sign of danger. For cortisol samples, I took samples at both baseline and after an acute stressor had been experienced to see what the stress level was of individuals as well as their physiological reactivity to stress. For all of these data sets, I used R to run generalized linear models, and factors in the AIC selected model were further investigated using ANOVA or chi-square. For the behavioral dataset, I used a principal components analysis to determine which aspects of swimming activity were most important. For mortality results, free embryos that had encountered perlids had significantly higher mortality, so perlids were definitely acting as a sturgeon predator. Unexpectedly, there was also a few mortalities in the isonychiid treatment. Turns out this is because isonychiids are facultative predators, so they can switch from filter feeding to preying on sturgeon if they want to. For cortisol results, I saw this beautifully clear pattern where cortisol levels correspond to the amount of predation being experienced. So baseline and post-stress cortisol was very low for the control treatment, and for the isonychiid treatment, the baseline cortisol was slightly higher, the reactivity to stress was also slightly higher. And then sturgeon that had encountered perlids had the highest baseline cortisol and the highest reactivity to stress. For the behavior results, I conducted 2 different principal component analyses, looking at the behavior of the fish before and after the alarm cue odor was added. For the sake of time, I'm just going to highlight PC1 from each of these analyses and summarize those results for you. So for the first analysis before odor was added to the petri dish, I saw that larvae that had encountered isonychiids were significantly less active, and this corresponded to lower mean PC1 scores. I also saw that after alarm cues were added, the larvae that had encountered isonychiids avoided the center zone of the petri dish more. That's probably because the alarm cue odor was added to the center of the petri dish, so they were reacting more to that alarm cue if they had encountered isonychiids. So the takeaways from this experiment are I saw that stress levels are proportional to the amount of predation on nearby conspecifics. And then I saw that sturgeon that had interacted with isonychiids had more behavioral alteration. So those are the ones that really behaviorally stood out from the others, with lower activity and more avoidance of alarm cues, even though they did have very low predation in that treatment group. One possibility might be that encounter rate with the aquatic insect larvae is important. So the fish that had encountered isonychiids frequently were pretty likely to survive until the behavior trials happened. So their behavior was probably influenced by all of those encounters. Whereas the fish that encountered perlids frequently were more likely to be eaten by the perlids before the behavior trials. So when understanding the consequences for sturgeon-free embryos out in the wild interacting with aquatic insects, it seems to matter not just which aquatic insects they encounter, but how frequently they encounter them. And this is just a glimpse into a very fascinating ecological system. It's already well documented that aquatic insects are very vulnerable to changes in environment, like water quality and temperature. So, for lake sturgeon populations, they're not only experiencing direct effects of climate change, but also experiencing indirect effects through changes to the interspecies interactions with these invertebrate communities. And a next interesting research question brought up by this experiment is, what is it in the environment that causes isonychiids to switch from filter feeding to preying on lake sturgeon? And thank you to my funding sources, and thank you to you for listening.